A few years ago, I tripped over a log at my friend's cabin and nearly fell face first into a campfire. I managed to avoid burning my face, but I did sustain severe second-degree burns on my hands in the process. Now, as you might imagine, my hands were covered in large blisters and were extremely sensitive for some time, which made even the most basic tasks incredibly painful. Now, if you think that's bad, imagine having third-degree burns, but instead of just your hands like me, they cover the vast majority of your body, as well as the inside of your mouth and esophagus. This is what patients with recessive dystrophic epidermolysis bullosa, or RDEB, deal with every day. They have mutations in a gene that encodes a crucial skin protein called type 7 collagen, which acts as a sort of biological Velcro to attach the top two layers of the skin to one another. Without this Velcro protein, the skin is incredibly fragile and is prone to blistering at the slightest touch. So fragile, in fact, that it has been compared to the skin of a butterfly. And on top of all of this, these patients are incredibly predisposed to developing a form of skin cancer called squamous cell carcinoma, or SCC for short. Now, what's so interesting to me is that SCC is rarely fatal in the general population, and yet it's a death sentence for RDEB patients. So why is that? Well, at the most basic level, cancer is caused by changes in gene expression and the mutations that cause those changes. So the goal of my thesis research is to identify the key genetic differences that exist between RDEB SCC and general SCC in order to find out why it's so much more aggressive in RDEB patients. Now, to accomplish this, I will isolate DNA and RNA, the genetic material, from tumor samples of each type. And then I can read this genetic material using highly advanced sequencing technology, which essentially allows me to look at how every single gene in the genome is being expressed and identify where all the mutations are in each sample. Then I can write programs that will dig into this vast amount of data and pull out the key genetic differences, kind of like a miner digging and digging deeper into a mine to uncover rare gems. Now, these genetic gems will provide insight into why RDEB SCC is so much more aggressive and eventually lead to the development of new therapeutics that can target these unique characteristics that exist in RDEB SCC in order to slow or even stop the progression of this cancer. Overall, the goal of my thesis research is to improve the quality of life for people already suffering from an unfathomably painful disease by studying the way that cancer develops and progresses. Thank you.